Amphotericin B, a potent antifungal drug, has long been a mainstay in the battle against serious fungal infections. Yet, the precise mechanism behind its effectiveness has remained somewhat elusive until now. In groundbreaking research, scientists have finally unveiled the molecular workings that make amphotericin B a key player in antifungal therapy. Understanding fungal infections. Fungal infections, caused by various species of fungi, are a significant threat to human health. These infections can range from mild, localized skin infections to severe, life-threatening systemic infections. Fungi are diverse organisms that can infect different parts of the body, including the skin, nails, lungs, and bloodstream. The treatment of fungal infections poses a unique challenge due to the similarities between fungal and human cells. This makes it difficult to develop drugs that selectively target fungi without harming human cells. Amphotericin B, however, has shown remarkable efficacy against a broad spectrum of fungal species, making it a valuable tool in antifungal therapy. Mechanism of action of amphotericin B. Amphotericin B works by targeting the fungal cell membrane, a crucial component of fungal cells. The cell membrane is responsible for maintaining the integrity and functionality of the cell. By selectively binding to specific components of the fungal cell membrane, amphotericin B disrupts its structure and compromises its integrity. The key to amphotericin B's effectiveness lies in its unique structure. It is an amphoteric molecule, meaning it can interact with both hydrophilic, water-loving, and hydrophobic, water-repelling, environments. This property allows amphotericin B to insert itself into the fungal cell membrane, forming pores that disrupt the membrane's structure and function. Once the membrane is compromised, essential cellular components leak out, and the cell loses its ability to maintain homeostasis. This disruption ultimately leads to cell death, halting the progression of the fungal infection. The role of amphotericin B in antifungal therapy. Due to its broad spectrum of activity against various fungal species, amphotericin B has proven to be a highly effective treatment for serious fungal infections. It is often reserved for severe, life-threatening systemic fungal infections where other antifungal agents may be less effective. Amphotericin B is particularly effective against fungal species that have developed resistance to other antifungal drugs. Its mechanism of action, targeting the cell membrane, makes it less susceptible to the development of resistance compared to drugs that target specific enzymes or proteins within the fungal cell. In addition to its direct antifungal activity, amphotericin B also possesses immunomodulatory properties. It has been shown to stimulate the immune system's response to fungal infections, further aiding in the eradication of the infection. Efficacy of amphotericin B in treating different types of fungal infections. Amphotericin B has demonstrated efficacy in treating a wide range of fungal infections. Its use spans across various medical specialties, including dermatology, pulmonology, and infectious diseases. The drug is commonly used to treat serious fungal infections such as invasive candidiasis, aspergillosis, cryptococcal meningitis, and mucormycosis. Invasive candidiasis, caused by Candida species, is a serious bloodstream infection that primarily affects immunocompromised individuals. Amphotericin B is considered the treatment of choice for this condition, as it has shown superior efficacy compared to other antifungal agents. Aspergillosis, caused by the Aspergillus species, is a fungal infection that primarily affects the lungs. It can be life-threatening, especially in individuals with weakened immune systems or underlying lung conditions. Amphotericin B, either alone or in combination with other antifungal drugs, is commonly used to treat invasive aspergillosis. Cryptococcal meningitis, caused by the Cryptococcus species, is a fungal infection that affects the membranes surrounding the brain and spinal cord. It is a significant cause of mortality in individuals with HIV/AIDS. Amphotericin B is a key component of the treatment regimen for cryptococcal meningitis, along with other antifungal drugs. Mucormycosis, caused by the Mucorales family of fungi, is a rare but aggressive fungal infection that primarily affects individuals with compromised immune systems. Amphotericin B is the first-line treatment for mucormycosis, as it has shown the best outcomes in terms of disease control and patient survival. Side effects and precautions of amphotericin B. Although amphotericin B is highly effective, its use is limited by its significant toxicity. The drug can cause a range of side effects, including kidney toxicity, infusion-related reactions, anemia, and electrolyte imbalances. These side effects can be severe and may require careful monitoring and management during treatment. To minimize the risk of toxicity, Amphotericin B is often administered under close medical supervision. Lipid formulations of the drug have been developed to reduce its toxicity while maintaining its efficacy. These lipid formulations have shown promise in reducing the incidence and severity of side effects associated with amphotericin B therapy. Alternatives to amphotericin B in antifungal therapy. While amphotericin B is a highly effective antifungal drug, its toxic side effects have prompted the search for alternative treatment options. Over the years, several new antifungal agents have been developed, offering alternatives to amphotericin B in specific clinical situations. Azoles, such as fluconazole and voriconazole, are a class of antifungal drugs that work by inhibiting the synthesis of ergosterol, a vital component of the fungal cell membrane. Azoles are effective against a wide range of fungal species and are often used as first-line therapies for less severe fungal infections. Echinocandins, including caspofungin and mycofungin, are another class of antifungal drugs that target the cell wall of the fungus. They inhibit the synthesis of beta-glucan, a key component of the fungal cell wall, leading to cell wall disruption and subsequent cell death. Echinocandins are particularly effective against certain types of invasive candidiasis and aspergillosis. Triazoles, such as itraconazole and posaconazole, are a subclass of azoles that offer broader-spectrum antifungal activity. They are often used as second-line therapies for fungal infections that are resistant to other antifungal agents. Research and developments in amphotericin B. The recent breakthrough in understanding the molecular mechanism of amphotericin B opens up new possibilities for improving its therapeutic profile. Researchers are exploring ways to enhance the drug's efficacy while minimizing its toxicity. One area of focus is the development of new formulations of amphotericin B that have improved safety profiles. Lipid-based formulations, as mentioned earlier, have already shown promise in reducing the risk of kidney toxicity and infusion-related reactions. Continued research in this area aims to further optimize the lipid formulations to enhance their efficacy and reduce their side effects. Another avenue of research is the development of combination therapies that include amphotericin B. By combining amphotericin B with other antifungal agents, researchers hope to enhance the overall efficacy of treatment while reducing the individual drug doses, thereby minimizing toxicity. Furthermore, the newfound knowledge of amphotericin B's mechanism of action can guide the development of novel antifungal drugs. Researchers can now target specific components of the fungal cell membrane more precisely, potentially leading to the discovery of new drug candidates that are more effective and less toxic than amphotericin B. Conclusion. The importance of amphotericin B in antifungal therapy. Amphotericin B has long been recognized as a key player in antifungal therapy. Its broad spectrum of activity and efficacy against serious fungal infections have made it an indispensable tool for clinicians. The recent breakthrough in understanding its mechanism of action provides valuable insights into how the drug works and opens up new avenues for improving its therapeutic profile. While the toxic side effects of amphotericin B have limited its use, ongoing research and developments aim to enhance its safety and effectiveness. The development of lipid-based formulations and combination therapies offer promising strategies to minimize toxicity while maximizing efficacy. As our understanding of fungal infections and antifungal therapy continues to evolve, amphotericin B remains a vital weapon in the fight against serious fungal infections. With the newfound knowledge of its molecular workings, scientists and clinicians can work together to optimize its use and explore new treatment options for patients in need. Thanks for watching.